Hey guys, this is going to be a rather detailed overview of sensory systems. Starting with the somatosensory system, which is involved in touch, pressure, temperature, and vibration. So the receptors in your skin are called cutaneous receptors. They're a type of mechanoreceptor. They react to pressures that change their shape that ultimately causes action potentials. They're of course associated with the cortical region and the post-central gyrus of your parietal lobe, which is called the somatosensory cortex. There are two types of cutaneous receptors. There are the free nerve endings, which are the least specialized, and these are what respond to changes in like temperature and pain. And then there's the encapsulated receptors, which are nerve endings that are encapsulated by connective tissue. So starting with your free nerve endings, they can detect changes in both temperature and pain. They're unencapsulated, and once again, they are the least specialized. They do express polymodality, so they can work as both thermoreceptors, mechanoreceptors, and nociceptors. Types include Merkel's fibers or Merkel's discs. Merkel's discs are discriminative light touch receptors, so that means that they react to low vibrations and deep static touch. The other type of free nerve ending is the hair follicle receptors that respond to light touch. And so you have just your free nerve endings that respond to pain, heat, and cold, your Merkel's discs that respond to touch, and your hair follicle receptors that respond to light touch. These are your goosebumps. So next is your encapsulated receptors, which remember are encapsulated by connective tissue. These are your Meissner corpuscles, your Pacinian corpuscles, and the Ruffini endings, or your bulbous corpuscles. Now Meissner corpuscles are located in the dermal papilla. They are light touch receptors, meaning they react to moderate vibration. The lamellated or Pacinian corpuscles are located in the reticular layer of the dermis, and they respond to deep and sudden pressure. So it has to be immediate, and if the pressure is chronic, they'll actually stop responding. So they only respond to sudden pressure. They're most sensitive to vibrations, and they also have really large receptive fields. Now, Ruffini endings are also called bulbous corpuscles. They lie in both the dermis and the hypodermis, and unlike the Pacinian corpuscles, they respond to the continuous pressure. So that was the somatosensory system. Now we're going to look at the visual system. So before we get into the visual system, let's go through the parts of the eye. So the ciliary body is the middle vascular layer that connects the iris to the choroid. And of course the choroid, also a middle vascular layer, is the layer that contains connective tissue as well as blood vessels. The cornea is the outer fibrous layer at the front of the eye. The iris is also a middle vascular layer that controls the size of your pupil. The pupil being the eye's aperture that allows light to strike the retina. Now the aqueous and the vitreous humors help maintain the eye shape. The aqueous humor is located in the anterior cavity. The vitreous humor is located in the posterior cavity. The sclera is the outer fibrous layer of the eyeball that's continuous with the cornea. The retina, which has the fovea, is the inner neural layer, right? The fovea is the very center. And then of course there's the optic nerve, which is also the inner neural layer where all the neurons are going to converge. So those are parts of the eye. Let's get into the visual system and how it works. And it all begins with light. What is light? Light is a periodic wave. And a wave is just a disturbance in a medium that transfers energy from one place to another, and this results in vibration. And so a light wave, in particular, is just made of traveling energy composed of oscillating electric and magnetic fields, and this is why we have what's known as the electromagnetic spectrum, or electromagnetic radiation. Now, because light is a periodic wave, you guys should understand and know the components of a periodic wave. You have the medium, which is represented by the horizontal line. Above the medium, the very top of the wave is called a crest. Below the median, the very bottom half of the wave is called the trough. The distance between the medium and the crest is the amplitude, as well as the distance between the medium and the trough. The wavelength is the distance between two crests or two troughs. We are most interested in the wavelength. Now, receptors in the eye detect certain wavelengths of light. So while white light is composed of various wavelengths, your brain sees the spectrum of visible light broken down into categories of red, orange, yellow, green, blue, indigo, and violet, such as light that's been refracted through a prism. So starting with your eye receptors, you have two types, rods and cones, and they're very different. What I want you to take away from this table is that rods are absent from the fovea. Cones are more common in the fovea. Rods also have a low sensitivity to detail, but they have a great sensitivity to light. That is why rods are considered the light and dark receptors. They also have no sensitivity to color. Cones, on the other hand, have a greater sensitivity to detail, and they are sensitive to color. And so what I want to actually make note here, and this might be mind-boggling for you, is that color does not actually exist outside of your mind. Light exists, energy exists, so wavelengths exist, and these wavelengths are interpreted by the cones and your brain as a particular color, and this makes your world more meaningful. And so that's kind of a mind-boggling thing, and some people ask me when I tell them this, then why do you have people who are colorblind? Well, if someone is colorblind, that means that there's an issue in their cone receptors being able to detect certain wavelengths of light, right? So because cones are the color receptors, there are three types of cones. There's the small cones that respond to wavelengths of 420 nanometers. There are medium cones that respond to wavelengths of 530 nanometers. There are large cones that respond to wavelengths of 560 nanometers. Rods only respond to wavelengths of 495 nanometers. That is why rods are your light and dark receptors. So once the signal passes through your pigmented cell layer, it then passes the neurotransmitters into the bipolar cells. The bipolar cells transmit the signal from the photoreceptors to the ganglion cells. Within the bipolar cell layer are what are known as horizontal and amacrine cells, and these are very important because they introduce lateral inhibition. And this lateral inhibition results in what we call a center surround. And center surround is just an area of a receptive field or a stimulus region. Now the reason why lateral inhibition is so important is because it is the capacity of an excited neuron to reduce the activity of its neighbors. So this results in a lateral direction of action potentials in the excited neuron that results in a greater sensory perception. And so that is the response of the horizontal and amacrine cells, is introducing that lateral inhibition for the greater sensory reception. So once the signal transmits through the bipolar cells, it then enters the ganglion cell layer. The ganglion cell layer is basically the final output neurons, if you will, found in the inner surface of the retina. The ganglion cells collect that visual information from the bipolar cells, and then that information is combined and interpreted as primary versus secondary colors in the primary visual cortex, or your occipital lobe. It is important to note that it is the ganglion nerves that converge to form the optic nerve. All right, so now we're into visual fields. The right eye receives signals from the left visual field. The left eye receives signals from the right visual field. The left visual field, if you remember, is interpreted in the right hemisphere, and the right visual field is interpreted in the left hemisphere. And notice that when you interpret an object, and now we're looking at the occipital lobe or the visual cortex, right? The object is interpreted as opposite and upside down. So you'll see that the green apple is on the occipital lobe of the left hemisphere, but it's now been turned upside down and around. So same with the pencil. So now we're moving on to the extrinsic eye muscles. These are innervated by your oculomotor, abducens, and trochlear cranial nerves. The inferior oblique elevates the eye and rotates it laterally. The inferior rectus depresses the eye. The medial rectus moves the eye medially. The lateral rectus moves the eye laterally. The superior oblique depresses the eye and rotates it laterally. And then the superior rectus elevates the eye. So now we're moving on to the olfactory system, which is your sense of smell. So the olfactory system is associated with odors. Odors are just volatile substances with a particular molecular weight. Gustation, or the act of tasting, is highly associated with the olfactory system. And so we're going to go into the gustatory system as well. So parts of the olfactory system that you want to be familiar with are the olfactory epithelium, the olfactory bulb, and of course the olfactory nerve. So olfactory transduction begins when the odorant interacts with the receptors in the olfactory epithelium. So the odorant has come up through the nasal cavity and has now interacted with the receptors in the olfactory epithelium, specifically with what are known as cilia and the dendritic knob. This sends an action potential down the neuron of the olfactory receptors that are located in this olfactory mucosa, and these olfactory receptors converge to form the olfactory nerve that innervates the olfactory bulb. Now, the olfactory region of the brain, they're numerous. The signal, of course, is relayed directly to the associated with cortical region. This is without being relayed through the thalamus, which you guys should know by now. The thalamus is like this gateway to all the associated cortical regions. Olfaction is weird because it doesn't actually use the thalamus. The signal is directly relayed to the associated complex. Now, the olfactory system innervates the amygdala, which is involved in emotional responses. It innervates the hippocampus, which is involved in memory. And this is why you can have what are called odor memories. So, when you start smelling chocolate chip cookies and it makes you think of your grandmother's cooking, It also innervates the orbitofrontal cortex and the hypothalamus, and of course the olfactory cortex of the temporal lobe, which is the conscious perception of smell. Hypothalamus is of course your major endocrine gland, and the orbitofrontal cortex is also the prefrontal region of your frontal lobe. All right, moving on to the gustatory system, which once again is your sense of taste, highly associated with your olfactory system. Gustation is the act of tasting. Gustatory receptors are located on the superior surface of your tongue, located basically in your taste buds. And of course, the projections at the very tail end of your taste buds are called papillae. There are several types of papillae. You have the valate papillae, which form an innervated V shape on the posterior surface of the tongue. The foliate papillae is located on the lateral surfaces of the tongue. These are mostly present in children, which is why you guys didn't see the foliate papillae on the model in lab. The fungiform and filiform papillae are scattered across the anterior 2/3 portion of your tongue, the filiform being the most abundant, and that is containing the tactile receptors instead of taste buds. So the gustatory pathway is stimulated by dissolved chemicals. Sensory messages are then relayed through the facial, glossopharyngeal, and the vagus cranial nerves to the medulla oblongata. The signal is then relayed to the thalamus and sent to the gustatory cortex, which, if you remember, is located in the insula just under the temporal lobe. Moving on to the auditory system, this is your sense of hearing. The auditory system processes and interprets acoustical energy. Your ear is divided into 3 regions: the outer, middle, and the inner ear. Your outer ear is composed of the auricle, the external auditory canal, and the tympanic membrane, or your eardrum. So the outer ear is going to collect acoustic energy and direct it through the tympanic membrane, and this causes the eardrum to vibrate. Now we're moving on to the middle ear, which extends from the tympanic membrane all the way to the oval window. Within the middle ear, you have these 3 auditory ossicles that transmit that sound wave from the eardrum to your oval window. That is your malleus, your incus, and your stapes. That is your hammer, anvil, and your stirrup. Now the pharyngotympanic, or the eustachian tube, connects the middle ear to your nasopharynx, and this equalizes pressure between the ear and the external environment. Now your inner ear is composed of the semicircular canals, the cochlea, and the vestibule. So once the sound wave gets transmitted through the middle ear, right, the malleus, incus, and stapes, to the oval window, the sound wave is now entering the inner ear. In the inner ear, there is extracellular fluid made of 2 components. Perilymph is within the outer bone of the inner ear, and the endolymph is within the inner membranous portion of the inner ear. So these semicircular canals house what are called hair cells. They're basically just at the base of the canal. At the base of the canal is this organ called the crista ampullaris, and it's the crista ampullaris that contains these hair cells. And on the tips of these hair cells are stereocilia. They're projecting upward into what's called the copula. And when these stereocilia get rubbed against by the copula, it causes a graded potential. And when that graded potential reaches a threshold, an action potential then occurs. Okay, that's how the signal gets transmitted. The cochlea also contains hair cells, but these are contained within the organ of Corti. And the organ of Corti extends into the endolymph of the cochlear duct. There's also what's called a basilar membrane. And when the basilar membrane vibrates due to the sound waves coming from the middle ear, the hair cells are going to bend, right, against that basilar membrane. And this causes potassium channels to open, and that's what causes depolarization to occur. Now the vestibule also contains its own set of hair cells inside what's called the saculi and the utricle. The stereocilia and the kinocilia of these hair cells project into the odothelic membrane, which contains these calcium carbonate rocks. And so when you tilt your head, it causes these hair cells to rub up against that membrane, that otolithic membrane, causing a graded potential. And then once again, when that reaches threshold, that will trigger an action potential. So those are all your sensory systems, and probably more detailed than you need to know, but I at least find it interesting, and so maybe you will too.